Hi, I'm Rita LaCaruso. I'm the PI of Project 2 for the Protect Center. Our project aims to understand the toxicologic mechanisms that underlie associations of environmental contaminant exposures and preterm birth. We're particularly interested in trichloroethylene and in the phthalates. Most of our work with the phthalates has focused in the past on metabolites of the diethylhexyl phthalate, but we're broadening that now with new people in the laboratory. So I'm standing right now in our tissue culture lab, and the tissue culture lab is where most of our work gets done because we're interested in generating in vitro models in which we can test some of these mechanisms.
Hi everyone, this is Sean Harris. I'm a research assistant professor in the Department of Environmental Health Sciences at the University of Michigan. I oversee the major laboratory and experimental operations in Project 2 of Protect. And in Project 2, we use a variety of in vitro, both primary tissue models and cell lines to model the biology of the placenta in order to gain greater understanding of the mechanisms of environmental toxicant impacts in the placenta and fetal membranes. Thank you.
Hey, my name is Trainee. I'm a professor here in the Department of Environmental Health Sciences. I'm collaborating with Professor Rita Loch-Caruso on Project 2 with this Protect program. So research in my laboratory focus on microbiome, biofilms, water quality, and human health. I have been collaborating with Rita for many years to study the interaction between bacteria and chemical contaminants and how that impacts the preterm birth. So I'm very excited to have this opportunity to work with this group to continue exploring the interaction between bacteria and chemical contaminants. I hope we, as a group, will be able to generate very exciting results and make some impact.
Hi everyone, I'm Kelly Bukulski, an assistant professor at the University of Michigan. I'm excited to be a new co-investigator on Protect Project 2, working with Dr. Rita LaCaruso. As a molecular epidemiologist, I help study the molecular effects of toxicant exposure on placental villus tissue.
Hi, I'm Kyle Campbell. I'm a PhD candidate at the University of Michigan. My primary advisor is Dr. Kelly Bukulski. I'm a molecular epidemiologist studying the role of the placenta in fetal maternal health. We collaborate closely with the Caruso-Harris lab to carry out this important work. My areas of interest include the cellular composition of the placenta and transcriptomics and epigenomic outcomes related to adverse pregnancy events.
Hello, my name is Lana Elkin, and I've been a Protect trainee since 2015, first as a PhD student and now as a second-year postdoctoral research fellow, working in Project 2, the toxicology component. My work centers around characterizing the effects of trichloroethylene exposure on placenta, and right now I'm using a rat model, and my work specifically looks at omics approaches such as RNA-seq, single-cell RNA-seq, and epigenetics.
Hey everybody, my name is Sammy Jaber. I am a senior in undergrad at the University of Michigan studying biochemistry. I currently work as a research assistant in the Caruso-Harris lab, and I primarily work under Dr. Harris, and I am still in training, so while I don't have many responsibilities myself at the moment, I'm learning to do things like running passages on cells when need be. I'm doing basic lab upkeep, and I've been using the comparative toxicogenomics database to get data on gene-chemical interactions so that we can potentially use that data for further experiments.